So I am indeed going to talk today about our efforts to build a theory for the evolution of ecosystems, and I'm going to describe what this even could mean. But I'll just start off by just reminding us that all around us we see lots of different kinds of ecosystems, and a common thing to many different kinds of these ecosystems is that they maintain just an incredible amount of biodiversity. And so in all of these ecosystems there's many different species, and so they all interact with each other, they self-organize, and through these kinds of dynamics thrive. And I would say that, of course, we know that this happens, but really if we want to understand quantitatively what is happening to the dynamics of these ecosystems, I would argue that in sort of the recent years, it's really in the microbial realm that we've been able to get unprecedented quantification of the dynamics of such systems. And so just very quickly, what these techniques involve is that you go out to some sort of an environment that you care about. You might care about the soil. You go and you scoop out a sample, you scoop out many samples, and you send it down through some DNA sequencing machine. I won't describe the details of that. You do some sort of analysis, and that analysis tells you what kinds of species there are, how many. If you do this over time, you can get the dynamics, and not only can you do this for soils, you can do this for marine ecosystems, you can do this for the gut, you can do this for different kinds of lab ecosystems if you want. And one thing that we've learned by doing this again is just like the macrobial ecosystems like forests and the oceans, it's also true in the microbial realm, it's even more so true that there are just an incredible amount of biodiversity. So just to give you some numbers, in every gram of soil you can see between 1,000 to 10,000 species, between 10 million and 1 billion cells of different microbes per gram of soil. These are just crazy numbers. And so because of the recognition of these crazy numbers, what we've also realized is that the classical paradigm of understanding ecology and sort of ecological dynamics, which was built on this intuition of low diversity, built on saying that if we understand what happens to one or two species, we can understand what happens to more, simply doesn't work anymore. And so in the recent years, what people, including us, have been able to come up with is to interrogate what happens to highly diverse ecosystems directly. And so just like in these previous kinds of work, what was really important is to think about these systems as systems that we can study using the methods of nonlinear dynamics. What is important or what has become important are different mathematical techniques, particularly from statistical physics and random matrix theory, that have let us probe what happens to high diversity ecosystems directly. And if this seems kind of mysterious, let me just very quickly tell you what kind of thing inspires us. So a very different problem which was facing people in the 50s and indeed the 40s was to understand the uranium nucleus. For various reasons people wanted to understand it. And so, this was really problematic because the uranium nucleus is very complex. It has, 92 protons, 138 neutrons, and so on and so forth. And so, this was a very very hard problem to solve. And so, Wigner over here came up with this basically genius move, which is that he said, okay, the uranium nucleus is really complex. I don't know how exactly to model it. What I'm going to do is that I'm going to assume that this complexity just means that I can replace the properties of the uranium nucleus with some random matrix. Okay? And this had no right to work, but what Wigner and since then others showed was that this theory that was based on imagining the uranium nucleus and its couplings as a random matrix did pretty well, especially well to predict one of the problems that people were interested in at the time which was to predict the energy level spacings of the uranium nucleus. So the details are not important here, but you can sort of figure out what are all the different energy levels of the uranium nucleus, and in particular you can ask for the spacings or the differences or the gaps between these different energy levels. And so here is, for instance, what that distribution of these energy levels looks like. Over here in this histogram in the back is data, and you can see in this curve over here, this is a random matrix prediction that came from Wigner. You can see it's a pretty good match. We understand many details since then of why this works well, etc., etc. And so what we've been trying to do over the past 5 or 10 years is to import these kinds of ideas into studying ecological systems. Okay, and so just to give you a very quick caricature of how we are able to do this, what we imagine is that we imagine models where there are species, there are many species, and there are different resources, different kinds of metabolites that these species need to consume. And what you write down are a large number of systems of equations, both which model the dynamics of the species and those that model the dynamics of these resources. And these two equations, these two sets of equations are coupled to each other, and they contain some pretty simple processes. For the resources, of course, the resources are supplied at some rate. They're depleted by the different species around them. That's how species grow. And of course, it might also be possible, which is especially true in the microbial realm, is that different species, when they consume resources, are also able to secrete other things as sort of metabolic byproducts. This feature is called cross-feeding, and so you can put that in as well. And so different species will grow by consuming resources, and then they might be paying some sort of maintenance cost to be able to work with these, right? So the details of the models are not so important. The important thing I want you to take away is that there are really a very large number of species, which means that this species equations are not one equation. There's a large number of equations, one for every species. For resources, again, there's not one resource, there are many different resources, and so there's a large number of equations for the resources. And typically when we say highly diverse in statistical physics, what we mean is that we're interested in a limit where the number of species and the number of resources go to infinity. And so for those of you who are not used to this notion, it's actually true that when these numbers go to infinity, things become easier to solve, etc., etc. But the important thing that comes up when you're trying to write down these models is, just like sort of Wigner was sort of worried, how do you figure out and choose what all the different parameters are? For instance, the parameters in these models could be parameters that have to do with which species consumes which metabolite, which species produces which metabolite. We don't have access to all of this information. And so what we do is borrow a trick from Wigner from the 50s, which is just like Wigner was dealing with the complex uranium nucleus and replaced that complexity with randomness. We take the complexity of metabolism, the complexity of these different ecological dynamics, and we replace it with a random metabolic matrix. Okay, and so by doing this, what we've been able to show over the last few years is that this can help us describe the statistical properties of the dynamics of these ecosystems, just like Wigner was describing the statistical properties of the uranium nucleus energy level spacings at different scales of ecological complexity. So we've shown that this can work to describe the dynamics of ecosystems that you bring to the lab, so like simple laboratory microcosms, but also to describe the dynamics and properties of different gut microbiomes. These are microbiomes that we have in our gut that help us digest our food, but then all the way up to marine and planetary scale ecosystems. Okay, my interest here today is, usually I would have given you a talk about one of these things, but my interest here today is not to give you a talk about one of these things, but to give you a talk about what is not in any of these models, which is a big caveat, which is that all of these models ignore the basic evolutionary process that has created and sustained the diversity of these species. Okay, and so this is actually a big challenge. And the challenge really is not to understand how the ecology of many species works, but instead to understand how ecology and evolution work together. And the reason this has been a big challenge in the field of ecology and evolution is been that these two processes have usually been studied separately. If you go to an ecology and evolution department in the US, they are one department, but there are ecologists who work studying ecological dynamics who pretend evolution doesn't exist. There are evolutionary biologists who pretend ecology doesn't exist. This is a simple caricature, but this is not far from the truth in my experience. And so really the reason that this is the case is not because people are stupid, it's because actually this is a very challenging theoretical problem. We don't know what we should expect when the processes of ecology and evolution run together. And so, I'm not claiming that we solved this problem, but I want to tell you in the second half of my talk about some recent work that we've been doing to take the first steps towards trying to incorporate evolution into ecological models. Okay, and so what I'm going to tell you about is we're going to play a simple game. We're going to start with a simple evolving population. I'm going to tell you about what the standard evolutionary paradigm of population genetics is. And we're going to ask what happens to this paradigm when you add a diverse ecological community of the kind that I was just telling you about. Okay, and so before I do that, I have to tell you at least something about the simple evolutionary paradigm. So the classic paradigm, the paradigm of population genetics, asks many questions. One of the simple questions, the classic cornerstone of the field, is you start off with a population of many, many individuals. Let's imagine that all these individuals are what we'll call wild type individuals, and into this population you introduce a new individual, which is a mutant. So you can imagine this mutant comes up from one of the wild type individuals that reproduces and mutates. And one simple question that you might ask yourself is that if I wait a long time, what is going to be the fate of this mutant? And there are really traditionally two answers. One is you wait a long time and this mutant goes extinct. You go back to the same picture that you were with, it's only wild types remaining. Or you wait a long time and this mutant does what is called fixes. It fixes in the population. Now all you see in the population are just these mutant individuals. You don't see wild types anymore, right? And so in some sense, when you ask the question, what is the fate of the mutant, you're asking what's the probability that this mutant goes extinct versus fixes, okay? And the classic way that people have tried to answer this question is to realize that these mutants have stochastic dynamics, which is to say that if I plot the frequency of this mutant, which is the fraction of the mutant individuals in the population, and I track it over time, there are many, many realizations that might happen because these dynamics are stochastic. There's birth and death events, which are random, and the randomness of these birth and death events results in some trajectories of mutants that go up all the way and fix, and some other trajectories that don't go up all the way that go crashing, and when you get frequency 0, the mutant's gone extinct. And the classic idea, and of course the mutant starts off here at time 0 at some very low frequency, usually one individual in the entire population. And so usually the classic way of sort of describing what happens to these dynamics mathematically is to solve what is called the Kimura equation. I don't want to go into the details of the equation, but the idea really is that this equation captures these two processes, one of selection, which just says, okay, the mutant grows better than the parent by some amount, which you call S, which is a selection coefficient. And of course, this process is not deterministic. There's a stochastic component. There's a noisy component which captures the stochasticity of the birth and death process. And so one of the things that we learned by solving this equation and by sort of studying it, this is sort of classical work done by Kimura in the 50s, is that there are really two different regimes. At low frequency, when the mutant is very, very rare in the population, stochasticity dominates. It's really the stochastic part of this, of the dynamics of the mutant that matter. But if the mutant manages to get up to some critical frequency, then actually selection dominates. And if this mutant happens to be fitter than its parent, it's almost guaranteed to fix in the sort of population. And so you can solve this equation and ask answer what is the fixation probability of the mutant. Okay, and so the dogma sort of classically in the field has been that this equation and this process, which contains only the parent and the mutant, that is what was there in this picture that I showed you, describes very well a very wide range of biological situations. And so what I want to tell you and what I want to remind you is that this is very different from the picture that is going on in nature. The picture that's going on in nature is not that there's just the parent and the mutant and they're slugging it out. It's that the parent and the mutant are actually embedded in a diverse ecological community. And so if we want to study the processes of population genetics, we ought to do it in this context, embedded in these diverse ecological communities. And so that's exactly what we did, which is that we asked, can we write down and describe a very general theory of what would happen to the parent and the mutant when they're embedded in this big community soup? And so really, you know, what I mean when I say general is that we're going to imagine again that there's a large system of equations we need to solve, one for every species in this community, and these equations have two components. One is the component by which these species or strains grow, and the other, this component of noise, which captures the stochasticity of the birth and death process that I was telling you about. And what we have and what we've been able to do is to come up with, in statistical physics language, what is called an effective theory of the dynamics of this mutant embedded in a diverse community. And when I say that this effective theory is universal, what I mean is that this G sub I, this growth function, how these different species grow, can be your favorite ecological model. So it can be any model. And so it can indeed also be a complicated model of the kind that I was showing you a few slides ago, where there are species, there are resources, species compete for resources, etc., etc. And you can imagine that there's one equation for all these different species in the sort of community. There's also these kinds of equations, one for the parent and one for the mutant. And you can imagine that the parent and mutant are similar to each other by some metric that you might devise. And our goal is going to be to predict the dynamics of the frequency of this mutant in the soup of this community. And so when I say the frequency of the mutant, I mean what fraction of the sum of the parent and mutant population is the mutant. So if this frequency is 0, everything is the parent. This frequency is 1, everything is the mutant. If this frequency is 0.5, it's half mutant, half parent. And so our goal is really to write down some sort of equation and study the dynamics of what this mutant's frequency does over time, just like the analog of the Kimura equation, which didn't have any ecology. Okay? And so my claim is that we can do this, and we can do this with a technique which is called dynamical mean field theory in statistical physics. I won't belabor you with what the details of that theory are and how we go about doing this, but I'll just tell you what the punchline is. And the punchline is that despite the ecological complexity of the community, despite the fact that there are a very large number of species, what you find is that compared to the Kimura equation, which just had selection and some effective population size, you need only one additional parameter to capture the complex feedbacks due to the community and their effect on the mutant. And so what I mean by that is that you can take this entire picture where there's a mutant and parent with a large number of species in the community, perform this procedure, and end up with just an equation which describes to you what the mutant does. And the effect of the community is just some simple single parameter that suffices to capture it. And so you end up with an equation that looks like this. And really what I want to point out to you is this one additional parameter that we need. So just, you know, the two parameters, which are the old parameters that we cared about, is S, the selection coefficient, how much faster does the parent go, the mutant grow compared to the parent. N, which is something called the effective population size, which tells you how much stochasticity matters. If you have larger populations, stochasticity matters less. If you have smaller populations, it matters more. And then really there's this one additional parameter which we call eta, which captures what we call the ecological feedbacks due to all of the other species in the sort of community. And so really the lesson is that there's this one simple parameter family that captures these ecological dynamics, and you can ask now, does it actually make a difference? What are the consequences of including ecology compared to this classic Kimura picture, which just had the parent and the mutant? And the answer is there is an important consequence. And to see that consequence, what we will do is we will plot the classical curve that people care about, which is the fixation probability. To remind you, this tells you how frequently, how many trajectories the mutant will fix in the sort of population, as a function of this parameter S, the selection coefficient, how much the mutant is better than the parent. So if it's 0, the mutant and the parent grow equally well. If it's positive, the mutant grows better. If it's negative, the mutant grows worse. And so just to remind you, the Kimura formula, which you get by solving the Kimura equation, which is a no ecology formula, it looks, if you plot it, something like this. So you know, like the fitter and fitter the mutant becomes, gradually the fixation probability goes up. And eventually, of course, if the mutant is very fit, you almost always fix. In the presence of ecology, with this model that I was showing you, you get a curve that looks like this blue curve over here. And just to sort of tell you in the last 30 seconds that I have, there are similarities and there are differences. And the differences are the important thing I want to point out. So of course, if the mutant is worse, of course the mutant is not going to fix. These are deleterious mutations. But really, there's this important regime in red where the mutant is actually beneficial. It's actually better than the parent, but its fixation probability is significantly suppressed compared to the absence of the ecology. And of course, if the mutant is eventually very, very good, then of course there's no surprise that it'll almost always fix. So compared to this gradual picture, in the presence of ecology, there's an almost switch-like picture. Below some critical fitness, you don't fix. Above some critical fitness, you do fix. And it turns out that this critical fitness is related to these ecological feedbacks that I was talking about. So with that, I'll end. The takeaway that I want you to have for this talk is that despite the ecological complexity of communities, which might make you think that this is a hopeless task, actually the dynamics of mutants in these communities can be surprisingly simple and predictable. And this predictability, statistical physics tells us, is not despite, but because of the high diversity of the community. With that, thank you for your time. I'm happy to take questions.
Wonderful talk.
Thanks.
So did you try putting 2 or 3 different mutants?
Yeah, that's an excellent question. So there is this regime, this is a regime of population genetics, which is typically called strong selection weak mutation. Mutants come up one at a time. There's another regime, which is a regime of clonal interference, where many mutants come up at the same time. This theory will fail for that regime. And actually, yeah, it's pretty hard to solve things for that regime. So this is something that we're working on right now.
So when you're looking at a community, there are multiple species.
That's right.
And so you really need to deal with it as a vector. If there are P species,
that's right.
Are you looking at the P vector, or are you looking at the mutant species versus the rest pooled?
So you are looking at the community vector, but what you're doing essentially is you're figuring out a way to coarse grain that vector.
So you're looking at one particular species and pooling the remaining, right?
Exactly, because you care about the mutant. What has
happened, but that's a major simplification, primarily because selection coefficients of the other species with which this species is interacting is also going to play a role.
Absolutely.
So you need to take S1, S2, so on up to SP, and that's a very hard problem.
Absolutely, yeah.
Yeah. How do you actually validate this?
Wonderful question. So we actually did manage to validate this. So one of the predictions of this kind of theory is that this one, that the presence of the community will actually result in what is called negative frequency dependent selection. Communities will suppress the fixation probabilities of mutants. And so what we've been able to do is to look at data sets from the gut microbiome, where people have actually been sequencing very deeply, different isolates. And so people have been able to track frequency dynamics in them. And so by fitting those frequency dynamics to models of this kind, you can fit what these different parameters are. And one of our predictions is that this parameter eta, which captures the ecological feedback, will always have a positive sign, which is to say it will always be negative frequency dependent selection. And that's indeed what we see. That's sort of prediction one that we've been able to validate. This was long time series, so you can make an independent measurement at every time for different patients. And so for all of these, we find that these eta measurements are consistent with a positive number. And the second prediction is that, what I didn't tell you is that this eta captures some coarse-grained properties of the community. And one of the properties that it captures is the diversity of the community. So what this predicts is that if there are sudden changes to the diversity of the community, you should actually change the strength of frequency dependent selection. And so in the same data set, they had patients who went who went through like big antibiotic treatments that killed off big parts of their gut microbiome. And so what we were able to show is that with no access to that information, just looking at frequency dynamics, we were able to detect those time points reliably. So that's another prediction that we were able to validate.
Yeah, so here one question.
Sorry, I don't. Yeah.
So I mean this term mutant which you have mentioned appears to be not so clear, because it has to relate to genotype or phenotype.
Absolutely, yeah.
Now, in a microbial population, there may be thousands of mutants.
Absolutely, yeah.
Some may have a positive value in the selection, some may not have, some have a negative value.
Correct.
So what do you actually mean by the mutant?
Yeah.
Because, and I always feel that the mutation and selection, that goes hand in hand for the evolution. So in a microbial population, there are always thousands or tens of thousands of mutations.
Absolutely.
And what are you talking about? Which mutant you are talking about?
Yeah.
Can you please specify?
Okay, wonderful. So I heard 3 questions there, so I'm going to respond them in the order in which I remember them. So first was the question of what is exactly a mutant here and how do you reconcile it with genotype, phenotype. I think it's absolutely true that we don't have a good understanding of what genotype, phenotype maps are. So in the model, there's a very good sense that we have of what a mutant is. The model only deals with phenotypes of different strains. That's their ability to consume resources. And so in our model, when we have a mutant, what we have is we take a parent's phenotype, that is, it's some vector by which it's able to consume different resources, and we make a small mutation, which is to say we ask for a vector that's similar to this vector but not exactly the same. And so that exactly is it's a phenotypic mutation. What exactly this means in terms of genotypes, we don't know, but that's because we don't have a good understanding of exactly what the genotype phenotype maps look like. That's answer 1. Question 2 was about the fact that there are thousands of mutants in a community. That's again absolutely true and goes back to this question that was being asked. So it's true that many microbial populations are in the regime of clonal interference, and so what we view this as is these models didn't even have the community to begin with, and so this we see as sort of the first step towards going and describing the dynamics of clonal interference. So I completely agree that that's where we need to go, but we're not there yet. And then the third question was about which mutant we're talking about here. I think that's an important question because one of the advantages and maybe disadvantages of an approach like this, which is theoretical, is that we are agnostic to a lot of the chemical details of what exactly we are talking about. So the advantages, I think, first I'll say the disadvantages. The disadvantages are that it's very hard to describe any particular system. If you want me to describe what is happening in the dynamics of your gut or my gut or soil outside in the IISc faculty hall, I can't do that using this modeling approach. But the advantages are exactly that if you want to understand very generically and at a conceptual level what can be possible or what we typically expect, that's exactly what this approach is useful for. So I think the question that this is trying to answer is, typically what kinds of effects of mutants would you expect to see?
So this ecological feedback is a time-independent parameter?
Oh, great question. So typically it is time dependent. But in this case, you get a remarkable simplification because of these 2 regimes of stochasticity dominated and selection dominated that I was telling you about. So it turns out that it's really only the early dynamics of the mutant that matters. And if for various reasons that I can describe offline, and so because it's the early dynamics that matter, you can build a very good approximation of and replace this time dependent parameter with a time independent one.
I see. Because I was just thinking about viral evolution in the context of the immune system.
Absolutely.
And there are a lot of quasi-species, but their evolution is going to depend on the antibody response, which will be completely time dependent.
So I don't think this will be a good model to describe those kinds of dynamics.
Akshit here, very nice talk. A quick question. Is there any way to understand intuitively, perhaps I missed it, why the ecology always imposes a negative feedback?
Yeah, great question. So really the idea is that there are 2 kinds of ways that parents and mutants interact. One is of course directly, and the other is indirectly through all the different paths in sort of community. So it turns out that if you have these kinds of competitive interactions, you end up getting an enemy of my enemy is my friend effect. So you end up getting situations whereby the mutant and the parent are kind of competing in the same niche, and so if the mutant is kind of doing better from the perspective of the community, this is just one niche, and so the parent does a little bit worse. So effectively, you kind of get this negative frequency dependent selection.
So last question. Very interesting. But here we are saying mutation occurs in one species and doesn't affect other species. But in real life, mutation can jump from one species to another.
Oh, you mean through horizontal gene transfer?
Yeah.
And
So
stuff? Absolutely.
have you looked at that, and how would that matter?
No. So these are, I mean, like I said, we view this as the first step, and these are absolutely the kinds of things that we want to play with in the future.
So your talk generated a lot of interest, you can see, but we have to close it. Thank you very much. I thank all the speakers and the audience. Thank you so much.